Hi everyone, welcome to week 4 of lab. Happy Wellness Day. This video is replacing lab for this week 4, so everyone is getting attendance points. Just expected that you are watching this video and still doing all of the things required for lab. All right, I will share my PowerPoint. Today, we are going to be talking about cell membrane transport. Here's a schedule of what we are going to be covering. So I will do a short lecture. We will watch the data collection video that is on YouTube and also available to you on Blackboard. We're going to do a couple of case studies, and then we'll talk a little bit about your lab report and what's to come next week in lab. So starting off talking about cell movement and transport, we can do this either passively or actively, moving substances across a cell membrane, either passively or actively. If we're doing it passively, this is not requiring any ATP. This means that the molecules that are moving across the membrane, or sometimes there isn't membrane, they're just moving. This is movement of molecules from a high concentration to a low concentration. This is the direction that molecules naturally want to move. So this does not require ATP. Active transport, however, does require ATP, and this is moving substances from a low concentration to a high concentration. This is the opposite of the direction that molecules want to move, so they need energy in order to move that way. Passive transport, different types are diffusion, so we can have simple or facilitated diffusion. Osmosis and filtration are all examples of passive transport, not requiring any ATP. Again, substances are moving from an area of high concentration to a low concentration, and this movement relies on the natural kinetic energy of those molecules, and this can be either assisted or unassisted, meaning that the molecules can either just kind of freely flow across unassisted, or they need some sort of assistance, a carrier molecule, still no ATP required. For simple diffusion, again, this is passive, so molecules are moving from a high concentration to a low concentration, no ATP required. This occurs either with or without a selectively permeable membrane, and these molecules are just flowing right on by. They don't need any help. But with facilitated diffusion, this is the same concept. However, there is always a membrane involved because these molecules need a carrier molecule to get across. So an example of this would be glucose movement across a membrane. So the glucose that is in the extracellular fluid gets into the cell with the help of a carrier molecule, and that's what this picture on the side is showing. So still no ATP is being used in this process. Those molecules just need a little bit of help getting into the cell, and that is by way of a carrier molecule with facilitated diffusion. There are some things that can affect diffusion, the rate of diffusion. Some of these are straightforward, like distance. If you have to go a shorter distance, it's going to be faster. Gradient magnitude. So the gradient is the driving force for the molecules to move to one place to the other. And going from a high concentration to a low concentration, the direction of the gradient is going to be going towards the low concentration, a high concentration to a low concentration. So the higher the concentration of the high concentration in comparison to the concentration of the low concentration, the bigger the difference that is on either side of the membrane, or if there isn't a membrane on either side of the space, the greater the difference, the faster the molecules are going to move. The more the molecules are going to be encouraged to move from that high concentration to a low concentration. And we'll make a little bit more sense of this in a bit. Molecule size, smaller molecules are going to move faster. temperature, the hotter it is, the faster the molecules are going to move. Electrostatic forces, opposites attract, like charges repel. We saw this last week when we saw all those hydrogen cations accumulating in the intermembranous space in the electron transport chain. So all of the hydrogen ions were being accumulated in the intermembranous space, and that encouraged them to then move across that membrane into the matrix. Some more examples of passive transport here. Filtration. This is going to be the movement of fluid and or solutes across a membrane due to fluid or hydrostatic pressure. A good example of this is your coffee maker in the morning. Your coffee maker quite literally has a filter that you pour water in, and you have the coffee grounds, and the water goes through, and so do some of those coffee solutes. So that is filtration, and hydrostatic pressure is the force that's happening there. And our final example of passive transport, no ATP required, is the diffusion of water from a high to a low water concentration through a selectively permeable membrane. And this is osmosis. When we're talking about osmosis, we are strictly talking about the movement of only water across a semi-permeable membrane. So here in this little schematic, we have some sugar molecules in red and some water molecules in blue, and our selectively permeable membrane is that green stick there. And so we see that only the water molecules are going to move across that semi-permeable membrane because they are being pulled towards the side that has a higher concentration of the sugar molecules. So trying to balance out the concentration of water on each side. Excuse me. We're going to dive a little bit deeper into osmosis here. Osmosis, it can make perfect sense to you at first when you're learning it, but I promise it has a very high likelihood of tripping you up. So spend some time with this. It might make sense now, but if after lab you go to answer some questions and it isn't making any sense at all, don't freak out. That happens to everyone. Just go back and review it a little bit, and I promise it will make sense eventually. All right, before we talk more about osmosis, we need to talk about a solution and the components of a solution. So a solution is made up of a solute and a solvent. The solute is going to be the minor component in a solution, and that is going to be dissolved by the solvent. And the solvent is the major component, which is doing the dissolving. Both the solute and the solvent can be a gas, liquid, or solid. It doesn't always have to be a solid as the solute and a liquid as the solvent. It can be any of those three substances. But for example purposes, I think it makes the most sense to think about salt water, where NaCl, table salt, is the solute, and then the solvent is the water. So the salt is the minor component, the minor component of that solution, and then the water is the major component doing the dissolving. And this results in a solution. Now, if we were to take a cell and drop it into that solution, we would be able to talk about the tonicity of that solution and the cell. So tonicity is the difference in solute concentration between a solution and a cell. So this is specific to our bodies. And water diffuses towards the higher solute concentration. So this is osmosis here because we're talking about the movement of water. So we can describe solutions as either hypertonic, hypotonic, or isotonic in relation to the cell that is inside of the solution. The solution is what we are describing as hypertonic, hypotonic, or isotonic in relation to the cell that is inside. Okay, diving in a little bit deeper. Let's start off talking about isotonic. Iso means same, tonic, tonicity, same concentrations. Equal concentrations on both sides of that cell membrane. This means that water isn't going to be moving inside of the cell or really outside of the cell because there's equal concentrations of solutes inside of the cell and outside of the cell in that solution. And since water wants to move to a higher solute concentration, neither of those concentrations are higher, they're the same. It's isotonic, so the cell is going to keep the normal shape. Water isn't going to be moving out of the cell, making it smaller, or moving into it, making it bigger. It's going to stay the same size. So this means that the extracellular fluid concentration is equal to the concentration of the intracellular fluid. And here's a picture here just to explain this, showing that yes, water is still moving a little bit, but it's not changing the shape of the cell. Hypotonic, again, we're talking about the solution. So there are fewer solutes in the solution than there are inside of that cell that we dropped into the solution. Since water wants to move to a higher solute concentration, the cell will gain water and it will swell and burst. In the body, we call this hemolysis when we're talking about specifically red blood cells. So hypotonic, there are fewer solutes in the solution than there are inside of the cell. Water wants to move to a higher solute concentration, so water is going to be moving into that cell because there are more solutes in the cell than there are outside. The solute concentration in the extracellular fluid is less than the solute concentration in the intracellular fluid. So that's hypotonic. And now hypertonic is going to be, again, we're talking about the solution. So there are more solutes in the solution than there are inside of the cell. In this case, cells will lose water and shrivel in the body. We call this crenate. Again, because water wants to move to a higher solute concentration. So since the cell has less solutes than the solution that it is being placed into, the solution is hypertonic in relation to the cell. The cell is going to lose water, again, shrivel and crenate. The concentration of the extracellular fluid, the solute concentration of the extracellular fluid is greater than the solute concentration of the intracellular fluid. Okay. In lab, if we were in person, this is the experiment that we would be doing together. What we did is we used dialysis bags to serve as our cells. So we fill up 3 dialysis bags with a 10% solution, and we clip both ends. We weigh each bag initially to get a baseline weight for them. And then we place each of those 10% solution dialysis bags into either a 0% solution beaker, 10% solution beaker, or a 30% solution beaker. Let that sit for 45 minutes, come back, reweigh the bags, see where the water flowed. Here's a video for us to watch.
Okay, so we have our 3 dialysis bags data.
Actually. I have to share a different screen. Excuse me, this is giving me a hard time right now. Okay, here we go. So the data that we give you in this video, go ahead and use this for your lab report, that table that's on the first page.
Okay, so we have our 3 dialysis bags here that are empty. We're going to fill each bag with a 10% salt and water solution and then weigh them. Once we're done filling all our bags, we'll weigh them. Now we're going to take each one of our 10% solution bags and put them in a respective beaker. So we'll have one 0%, one a bag fully submerged, one is in a 10% solution, and our third will go in a 30% solution. And now we'll sit and let these wait for about 40 minutes. So after about 40 minutes, we're going to re-weigh them and record their weights.
At the very end of that video, here, there is your data to enter for into that graph on your lab report.
Your skincare routine?
Oops. Okay, now that was the experiment that we did. And I'm going to share my PowerPoint again now. Okay. So this is a visual example of what we just did in that video. We took a 10% solution, quote unquote, cell, which is really the dialysis bag, and we placed it into, we took three 10% solution cells and placed them into either a 0% solution beaker, which would mean that the solution is hypotonic in relation to that 10% solution cell. We put that 10% solution cell also into a 10% solution beaker, meaning that the solution in the beaker was isotonic in relationship to that cell. And we also put that 10% solution cell into a 30% solution beaker, meaning that the solution inside of that beaker was hypertonic in relation to that cell. And I will let you guys decide what happens to the cells there in the beakers with the question marks, whether they're going to swell, remain the same size, or shrink. So that was all passive transport, not requiring any ATP at all whatsoever, with diffusion, filtration, and osmosis. Passive transport. Now we're going to talk about active transport. And this is transport that requires ATP and moves molecules against a concentration gradient, meaning they're going to be moving from a low concentration to a high concentration. This is not the direction that molecules naturally want to move in, so they need energy, ATP, in order to be moved against that concentration gradient. Or high to a low concentration or pressure. An example here would be the sodium-potassium pump. We'll talk about that in the next slide. And this is also going to be bulk movement of materials into and out of the cell via exocytosis and endocytosis. Here's our sodium-potassium pump. Requires ATP for this to happen. You learned in lecture that the sodium-potassium pump pumps 3 sodium molecules out for every 2 potassium molecules in. This is a critical system for cell homeostasis. I think this schematic on the side here gives a good understanding of how this works. So we know that there's a really big K on the inside, meaning that naturally at rest, most of the potassium in our bodies is going to be inside of the cell. There's a really big Na outside of the cell, meaning that naturally at rest, most of the sodium is going to be outside of the cells. And that's representative of their concentration gradient. So there is less sodium inside of the cell than there is outside of the cell. So naturally, sodium is going to want to move in. With potassium, there is more potassium inside of the cell than there is outside of the cell. So naturally, potassium is going to want to move out. And that is simple diffusion, no ATP. So we have this pump to pump sodium and potassium in the opposite direction. So we have this pump to keep on pumping potassium in and keep on pumping sodium out so that we can maintain our membrane potential, resting membrane potential. And also, the bulk transport of materials across the cell membrane also requires ATP. This is done by exocytosis and endocytosis. Exocytosis is moving fluids or particles packaged in vesicles across a cell membrane outside of the cell. So that picture shows that that vesicle is going to fuse with the cell membrane and then just release whatever particles are within that vesicle out into the extracellular space. And then endocytosis is the same but opposite. This can be either phagocytosis or pinocytosis. So this is when the cell membrane engulfs large particles and small fluid particles. So when the cell membrane is going to engulf larger particles, this is going to be phagocytosis, cell eating, and then the smaller fluid molecules, when those are engulfed by a cell membrane, that is called pinocytosis or cell drinking. And again, both of these processes require ATP. They are active transport. Okay, so at this point in my lab session yesterday, we did breakout sessions for this, but at this point, I ask you to maybe pause this video and just go ahead and read through those hyponatremia case studies on page 2 of your lab document. Try and answer those questions 1 through 4 on the hyponatremia case study, also on page 2 of the lab document. So go ahead and pause this, and then I will go over those answers here. Okay, let me grab my document. Okay, for hyponatremia, after reading those case studies, what sort of environment, either hypertonic, isotonic, or hypotonic, does consuming excessive amounts of pure water create in the body fluid that surrounds your cells? So if you're drinking pure water with no solutes in it or anything, just H2O molecules, this is going to create a hypotonic environment in the fluid that surrounds your cells. Remember, with osmosis, we are always talking about the solute, or sorry, excuse me, the solution, the solution that the cells are inside of. Excuse me, my dog is crying. Sorry about that. So that is going to be creating a hypotonic environment for the solution that the cells of your body are sitting in. What effect would this have on your cells? Kind of the same question as number 2. Explain the consequence of such an environment on the cells of the body. So if it's a hypotonic solution, if we set a cell into a hypotonic solution, where is the water going to flow? The water is going to flow into the cell because the solution is hypotonic in relation to the cell. The water is going to want to flow to where there's a higher solute concentration. So the water is going to flow into the cell. They might swell and then lyse. Lyse is the correct word that we would use for what's happening in the body. Number 3, why do you think doctors administer a saline solution instead of pure water to dehydrated patients? I think these case studies answer that question for you, because we don't want to create a hypotonic environment for those cells of our body. We want it to be isotonic. We don't want cells to be gaining water and swelling and bursting and lysing. No, we we want them to not change shape when we administer saline or whatever solution you're administering to the body. So we administer saline solutions so that we create an isotonic environment for the cells of our body. Google hyponatremia, define it, and discuss how it relates to the above cases. Hyponatremia is not enough sodium in the body. This can be either from not ingesting enough sodium or drinking too much water, drinking too much pure water, or maybe a combination of both. Okay, so that was the hyponatremia case study, not enough sodium in the body. Now on the next page of your lab report, you have a hyperkalemia case study. And I'm going to briefly go over that with you guys. Okay. Here's our hyperkalemia case study. A 72-year-old man with coronary artery disease, renal disease, and diabetes presented to the ER with swelling and shortness of breath. An acute myocardial infarction, which is a heart attack, was ruled out, and he was diagnosed with congestive heart failure. An angiotensin-converting enzyme inhibitor was started before discharge. 2 weeks later, he presented to the emergency department with fatigue, lethargy, and a critically elevated serum blood potassium level. Shortly thereafter, he suffered a cardiac arrest and died. So hyperkalemia means too much potassium in the blood. Question number 1 here is asking us to draw or describe the intra and extracellular concentrations of sodium and potassium of a cell at rest. So I will go ahead and draw that out for you. So here's our cell at rest. This is going to be backwards for you. Maybe I can... Aha, there we go. So here's our cell. At rest, we're going to see that there is a bunch of potassium inside of the cell. I don't know if Professor Wallace still uses this way of describing things, but the way that I learned it was someone asks sodium and potassium if they want to go to a party. Sodium says K, so they're in the party inside of the cell. And, potassium says K. I'm not sure if I said sodium or potassium there. Potassium says K, I'll go to the party inside of the cell. And sodium says, nah, I don't want to go to the party, so I'm going to be outside of the cell. And this is the concentrations of sodium and potassium in the cell at rest. We still have some sodium in the cell and a little bit of potassium outside, but naturally at rest we have a bunch of potassium inside of the cell and a bunch of sodium outside of the cell. Going back to our case study here. Question number 2. Though in a healthy person, the correct concentration of the ions is maintained both intracellularly and extracellularly, there is much movement of these ions taking place across the cell membrane. Describe both the active and passive transport that is occurring for sodium and potassium. I'm going to go back to my whiteboard here. Okay. So we just said at rest, there's a bunch of potassium inside of the cell, and there's a bunch of sodium outside of the cell at rest. So, with the information that we learned today about simple diffusion and also with active transport with the sodium-potassium pump, we can draw some conclusions here. So just simple diffusion-wise, with simple diffusion, molecules are moving from a high concentration to a low concentration. This is naturally happening without ATP all the time for these sodium and potassium molecules. So if we're moving these along their concentration gradient, passive transport, simple diffusion, no ATP, where the molecule is going to be flowing. From a higher to a lower concentration, that is going to be from inside of the cell to outside of the cell for potassium. And then for sodium, moving from a higher concentration to a lower concentration, that is going to be from outside of the cell to inside of the cell. And that is just simple diffusion, no ATP. This is just happening. This is the natural kinetic energy of these ions that's encouraging this passive movement. All right, so that's passive. What about the active? I'll use a different colored marker here. So for active transport, we have that sodium-potassium pump. So this is happening all the time, all of this passive stuff, but we also have this sodium-potassium pump here. That is, active transport requires ATP because it's moving these ions against their concentration gradient. So that means that with the active ATP required sodium-potassium pump, we're going to be pumping 2 potassium ions in for every 3 sodium ions that we pump out. All right, and this is active, requires ATP, and the rest in blue is passive, just happening along their concentration gradient. Whereas this here, the sodium-potassium pump, is happening against their concentration gradient, and that's why ATP is needed here. Let's go back to our case study. Question number 3, how would an elevation in extracellular potassium affect the resting membrane potential of neurons? What's our resting membrane potential of neurons? Negative 70 millivolts, small m, big V. And now I'm going to go back to my whiteboard to draw this out for you. So let's pretend like this is a neuron. Negative 70 millivolts is our resting membrane potential. I really hope that this isn't backwards for you guys, because now that I unmirrored my screen, it's backwards for me, and let's just hope that it's not mirrored. But in any event, what would happen to this negative 70 resting membrane potential if the extracellular concentration of potassium is increased? If all of a sudden, like in this case study, we have a case of hyperkalemia, too much potassium in the blood, in the extracellular fluid. What does that do to this concentration gradient here? This concentration gradient that normally there isn't much potassium outside of the cell. Normally this concentration gradient is very much encouraging the movement of potassium that is in the cell in a high concentration moving it along its concentration gradient to a low concentration outside of the cell. That's normally what's happening at rest. There normally isn't a bunch of potassium outside. But since in this case we have hyperkalemia, what's going to happen to this concentration gradient? Potassium isn't going to want to passively exit the cell as much. It isn't going to passively diffuse across the cell membrane because our concentration gradient isn't as strong. It isn't encouraging potassium to leave the cell as much because there's already a bunch of potassium out here. And so while all of this is happening, while the potassium isn't really leaving the cell quite as much because there's a bunch out in the extracellular space now, this sodium-potassium pump is still doing its job. So it's still pumping all of that potassium in, pumping sodium out. But also passively, potassium is just kind of hanging out in the cell and not really being encouraged to leave because that concentration gradient is almost disappeared. So that means that we're going to have an accumulation of positive charges inside of the cell. We're going to have an accumulation of positive ions, all these potassium, because they're not being encouraged to leave through simple diffusion because there's so much outside. What's going to happen to that negative 70 number is going to become more positive. And the more and more potassium that accumulates in here, the more positive it's going to be, the more, the closer that millivoltage is going to get to zero. And that means that the cell, since its resting membrane potential is getting closer to zero, that means that cell is going to become more excitable, more likely to depolarize, since we're getting that resting membrane potential is getting closer to that threshold, right? So I'll go back to the case study. And number 5, what do we think happened in our case study? So since we saw that the elevation in extracellular potassium affected the resting membrane potential of a neuron by making the resting membrane potential more and more positive because we have an accumulation of positive ions inside of that neuron, right? So the resting membrane potential is going to become more positive, closer to zero, making that cell more likely to depolarize and ignite an action potential. If that's the case for neurons, what about our study? This person had suffered from a heart attack because they had hyperkalemia. This same situation can happen to the excitable cells of your heart, the cells of your heart that start the action potentials. So if this happens to the cells of your heart, same situation as the neuron, the resting membrane potential is going to become more and more and more positive, making that cell more and more likely to depolarize. In the case of a heart, this can cause an arrhythmia because those cells are so excitable, they will just depolarize whenever. This makes it so that the heart doesn't beat as synchronously as it should, causing an arrhythmia, causing someone to have a heart attack and die. So that is what happened in our case study. Okay. Going to go back to my PowerPoint now. Okie dokie. And that is all that I have for you today. Now I know maybe those case studies were a little bit tricky, and some of the material that I covered in lab today might have been a little bit tricky for you. So please go ahead and send me an email or, reach out to Professor Wallace, the other GAs, the SI. We're here to help you. We want to see you succeed. We want you to understand this stuff because it's very fundamental material that you will need to know for your future classes. So we want to help you out. Let's take a look at the lab report really quickly before I finish this up. I don't want to share that. Okay. So here's our lab report. Questions 1 through 3 describe what happened in each dialysis bag and why this occurred. Be sure to include a discussion about tonicity. So earlier, I showed you that data collection video. Make sure you jotted down the information that was in that video for that chart up here for this. Okay. So go ahead and describe what happened in each dialysis bag, and you must talk about tonicity to get full points for questions 1 through 3. Number 4, how would the extracellular tonicity be affected in a person who is dehydrated? What effect might this have on their cells? So go ahead and think through that. It is possible for a person to die if they drink too much water without also replacing electrolytes. That's a question. Is it possible for a person to die if they drink too much water without also replacing electrolytes? What effect might this have on their cells? Refer to the hyponatremia case study. Number 6, describe why a person with hyperkalemia, too much potassium in the blood, will have neurons and muscle cells that are more excitable than normal. We just talked about that, so go ahead and maybe rewind this video if you need to and look at that again. And number 7, describe where sodium would accumulate and what effect would this have on water movement across the membrane if the sodium-potassium ATPase pump were to stop working. Go ahead and think through that. If you need to, rewind this video and rewatch some stuff. It will make sense. Just take some time. And then go ahead and read this case study and answer these questions on it. And finally, remember to provide a correct citation. I've had some questions. What do I do with all of this material that we learn in lab? Do I have to cite that? No. For our purposes, everything that we learn in lab and lecture is considered common knowledge. So anything that you cite is only extra sources that you use. And if you find yourself not finding any or not needing to look for any extra sources, go and find one anyway and cite it just so that you get that citation point, okay? Make sure that it ties in with what we're doing and you can actually use information from that citation for your lab report. It's not just a random reference. Okay, that's our lab report. Now, what do we have to do next week? So over the course of the week, make sure you take your week 4 lab quiz, so your membrane transport lab quiz, by this Sunday at midnight. Even though today was a wellness day, this is still treated like you're in lab, basically, so you still need to turn in that quiz and the lab report and everything like normal. So take that quiz by Sunday at midnight and work on your membrane potential lab report and send it to me before lab next week. That 10:29 a.m. strict deadline. So next week, is week 5. We're going to continue talking about neural and hormonal communication in lecture. And in lab, we are going to do a question and answer session for exam 1. I will probably make a Kahoot so that you guys can interact and it isn't just me talking at you the whole time. So that's next week, week 5. And then week 6, on the 15th, is your first exam. So how are you going to prepare for that? Hopefully you're all caught up and up to date on the lecture material. You've been attending lab, so that's great. Something that I definitely recommend you doing is hopefully you've been attending those SI sessions, and definitely also download those chapter review guides that Professor Wallace posts on Blackboard for you under the exam 1 material. Go ahead and do those. Try and do it without looking at your notes. I promise you they will help a lot. And if you're able to go through those without your notes, you should feel pretty confident going into those exams, right? Okay, that's all I have for you today. I hope you guys enjoy the rest of your wellness day, and I will see you next week.